Welcome to Histology Lab. Today we're going to study cartilage and bone tissue. Recall that cartilage and bone are both specialized types of connective tissue. In these labs, we're going to be having a guided tour of sorts where I take you through the lab guide. You can either follow along the video using your lab guide document, or you can just watch the videos and then review the lab guide later, looking at the slides using those offline links. Let's start by looking at cartilage. Recall from the self-study notes that there are three types of cartilage: hyaline cartilage, fibrocartilage, and elastic cartilage. Let's start with hyaline cartilage. This is the esophagus trachea slide. There are several slides in your collection that have the hyaline cartilage. In this esophagus and trachea, you can see this dark ring with cartilage. We studied this when we looked at the respiratory tract. So let's look at some of those characteristics of this hyaline cartilage. Here at higher mag, you can see that you have this band of cartilage. Each one of these little spaces used to contain in life a chondrocyte, which is the name for a cartilage cell. The space in which the cartilage lives, which is what you're seeing here, is called the lacuna. Many of those cartilage cells are gone. You can see some of them still intact, but some of them artifactually have fallen out or been torn. In any case, each one of these lives in its own little lacuna, and when they divide, you get sometimes these pairs or even quads of them. So there might be 2, 4, 8 isogenic pairs within these little areas. They make some more of their own matrix and push apart from one another. That's interstitial growth. They also make new cartilage cells here at the edge, where we have this dense connective tissue covering called the perichondrium. Hyaline cartilage has a perichondrium. So does elastic cartilage. Fibrocartilage does not have it. And hyaline cartilage in very specific locations, such as articular joints, does not have a perichondrium. But in this case, you do see the perichondrium. So this is a dense connective tissue layer, the innermost portion of which is chondrogenic. In other words, these cells are stem cells for growth of cartilage. So new cartilage cells can be made there. Moving along, let's look at the developing digit and nail slide. Here you can see a forming bone. There's some bone forming in the middle, and we'll look at that process when we look at bone in the second part of this lab. But the ends of it here, of all of these areas, are hyaline cartilage initially. So let's take a look at that. Here we are at medium mag. You can see that these are isogenic groups of cartilage cells or chondrocytes in this homogeneous matrix. And that's one of the ways that you're going to ID this type of cartilage over the other two types, is that the matrix looks very homogeneous in many cases. When I say homogeneous, what I really mean is that there's not visible fibers within there. And that's because the collagen is collagen type 2 here, and that does not form fibers. It forms fibrils, which are dispersed in a network. Another example of this type of cartilage, hyaline, can be found in the pig embryo slide. And that would be in these locations here and in this area here, which is the developing nasal conch. So let's zoom in on that and take a look at a higher mag at that developing hyaline cartilage. Here you can see that same homogeneous background with the chondrocytes sitting in their lacuna. And what you may notice is there's a lot of these dividing pairs, because this is still a growing cartilage. This is embryonic tissue, so the cartilage itself is still growing here. One more example is in the developing membrane bone slide, and here in the center is also another example of hyaline cartilage. Now this is a trichome stain, so it looks a bit different than the other ones we've looked at, the H&E ones. And here, the matrix appears this bluish color due to the staining of some of the matrix components. I believe it's hyaluronan and the collagen type 2. But the same characteristics are going to be present in terms of the morphology. In other words, the cells are going to be in groups or single with this homogeneous background. They just look a little bit different in terms of their color. Finally, we're going to look at hyaline cartilage in the epiphyseal plate of long bones. And this is the tibia slide. There's two of these. We have an H&E and a collagen stain. And the epiphyseal plate is right along here. So we'll zoom in on that a bit. So in this region, cartilage is making a template to form bone, and we'll examine that process when we look at the development of bone. As I said previously, that we'll do that in the second part of this lab. But for right now, as we zero in on this, we can see that we have again that homogeneous background, and here the cells are organized quite differently because of the development of long bone, but still the same staining characteristics with the chondrocytes sitting in their lacuna. You also have hyaline cartilage at the end of this bone on the articular surface. So let's move down to that portion, which is right here. And you can see that you have hyaline cartilage here. Again, that nice homogeneous background with the cells in their lacuna. And they are organized slightly differently here. You have some cells organized almost in little groups, kind of vertically, and then here they're more or less horizontally at the articular surface. Now what is missing here? This is one of those cases where hyaline cartilage does not have a perichondrium. So the articular surface is not covered by a perichondrium. You have chondrocytes all the way up to the edge. What does that mean for this cartilage? It means that it does not have much chondrogenic potential. So if this cartilage becomes damaged, as happens in various types of arthritis, the chondrocytes will die and the matrix will begin to degrade, and that's when you get the narrowing of the joint space as you lose this tissue. So let's compare this to the collagen-stained version of the tibia. So here's the epiphyseal plate with that collagen stain, and in this case, the cartilage appears in this kind of orangey red color, and the bone is this aqua green color. However, in spite of those changes in color, the morphology is still pretty much the same. You have that homogeneous matrix, and the cells are organized in the rows as the long bone is growing in this direction. Now let's switch gears and take a look at fibrocartilage. There's a couple of examples of that in your slide set. So let's zoom back out and look at this region over here. Now, fibrocartilage is typically a very transitional tissue between two other things, and one of its partners is typically hyaline cartilage, which you can see here, and it often transitions into a dense connective tissue, tendons, ligaments, things like that. So where you have this transitional area between those two different things, that's the fibrocartilage. And it's characterized by having a mixture of type 2 and type 1 collagen in its matrix. That contrasts a lot with the hyaline cartilage, which is strictly type 2 collagen. Now, how do you know that? Well, the type 2 collagen appears that homogeneous look, And the type 1 collagen is forming these thick bands throughout it. And you can see that very nicely in this collagen stain, which is actually the safranin O and fast green stain, because the type 1 collagen areas appear that blue-green and the type 2 appear the reddish color. So you have that kind of alternation. What's another indication or characteristic of that fibrocartilage? It's that the chondrocytes tend to line up in these rows because you have a directional kind of appearance as you transition from this tissue over here, the hyaline cartilage, to this tissue over here, which is the dense regular connective tissue. Another one of our best examples of fibrocartilage is in the pubic symphysis slide. So right here in the center, you're going to see a big chunk of fibrocartilage. So let's zero in on that. And again, you have that transition, and in this case, it's between these areas here where you have hyaline cartilage, and here you have the fibrocartilage between those two. And just like I said previously in the other slide, you have this mixture, this kind of fibrous appearance of the pink bands of type 1 collagen, because this is an H&E, so collagen will appear those pink bands, with the very purplish background of homogeneous type 2 collagen that you see in this hyaline cartilage area. So this is the fibrocartilage in here. And again, you have the cartilage cells or chondrocytes lining up in these rows in the direction of the bands of collagen that run through this, the type 1 bands. The final type of cartilage we're going to examine is the elastic cartilage. Now, you really cannot distinguish elastic cartilage from hyaline cartilage without a specific elastic stain. We're going to look at this slide of the epiglottis, which has been elastic stained, and just as we saw when we looked at general connective tissue way back in the beginning of the year, we're going to see the elastic fibers looking like black lines or fibers in the matrix. So here we go, jumping up in magnification, zooming back out again. This is your cartilage compartment here, the width of it, and you have perichondrium on either side and chondrocytes in their matrix in the middle. Now, at higher magnification, when we look at this, the matrix appears full of these black clusters or groups of elastic fibers throughout the matrix. In some areas you can see the fibers a little more clearly, like this. It's very characteristic, but again, the lacuna with chondrocytes in it, they look pretty bad here in terms of preservation, but you can see the lacuna and where the chondrocytes used to live, and the matrix, which is full of the elastic fibers. Now let's switch gears and talk about bone. This first sample is ground bone. And ground bone is not a section. What they did was they took a hunk of bone, they put it into acid and dissolved all the organic portions. So there's no cells, no collagen, that's all gone. All you've got left is the matrix that was calcified. So you've got calcified stuff. Then they put it on a lathe and they ground it really thin enough to put light through it. So this is really just a slab of calcified stuff. But in this orientation, it allows you to see the structure of how the components of what we call compact bone is put together. So bone, as you might recall from your self-study, has two main types, compact bone and spongy bone. So let's start with the compact bone by looking at this ground bone specimen. I'm going to zoom up on this, and you will hopefully notice that you have these lamellar kind of structures here. Some of them are whole and some of them are partial. And then you have some open areas as well. Each one of these lamellar structures is called an osteon. So an osteon is a cylindrical structure. These are cut in cross section, so you're seeing one plane through this cylinder. And it's made up of various lamella or layers of bone cells that were laid down. In the center, when it was alive, there was the vasculature and nerves, and that's where all the communication and nutrients and oxygen and everything came in and out. And in these black areas that look like smaller, let's go up at higher mag, these black, almost flattened disc areas is where the bone cells used to live. So this central area is called the Haversian canal, sometimes referred to as the osteon canal, since this is an osteon. These spaces are, again, just like when we looked at cartilage, these are going to be called lacuna, because these are the spaces that used to hold bone cells. They're where the bone cells used to live in this tissue. Not just any bone cells, though. There's actually 3 types of bone cells, if you recall from your self-study material. The cells that lived here are osteocytes. These are the ones that maintain the bone and communicate with one another. If you look very closely at high mag, you can see that there's this system of very small little canals where the cells that lived in here had cytoplasmic processes projecting out into these little canaliculi, which is what these little hair-like openings are called. And they reach out their cytoplasmic processes to one another and they communicate so that what's coming in here can be passed layer to layer to layer, all the way out to the outermost layer of the osteon. Sometimes you will see canals that appear to be lateral, and at the bottom and top of each of these cylinders, each of these osteon cylinders, they often have intercommunicating channels, and those are called Volkmann's channels or Volkmann's canals. Let's look at a different version of compact bone. This is kind of the diametrically opposite from the ground bone slide. In this case, it was put in a solution that got rid of all of the calcified components, and all you're left with is the organic components of the matrix. And the cells here are actually here. Now, the osteons are not as obvious in this compact bone, but still they can be identified. So let's zoom up a little bit and look at some features at medium and then high mag. So when you're looking at, this is a long bone that was cut in cross section. When you're looking at the compact bone portion, or the shaft of this bone, it has an outermost layer, which is called the fibrous periosteum. Let's zoom onto that. And you can see that's a dense connective tissue layer that covers the outside of this bone. And on the inner surface, next to the marrow cavity, this is artifactually separated, there is a single layer of cells that lines the inner surface here, and that's called the endosteum. Hopefully we'll find a better area of that if we look around. Here we go. Here's a better area. So here you can see the single layer of cells of the endosteum that lines the marrow cavity. Let's go back to looking at the compact bone itself and look for some osteons. As I said, these are not as easy to locate in this type of preparation, but you can in fact find them. So here is an osteon, and here's another osteon, and a portion of one here. So you can see at least 3 different lamella here. Here you can see 1, 2, 3 lamella. And you can see some interstitial bone as well between the osteons. And it has all of the same characteristics that we saw in the ground bone, but here the cells are actually present. Whereas previously in the ground bone, you just had the lacuna that you were looking at. You didn't have any actual cells. So you can see the haversian canal, the lamella, the osteocytes. Sometimes inside of this chamber, you will see, okay, so here's an example of something called a resorption chamber. So compact bone, matter of fact, all bone, spongy bone and compact bone, remodels constantly. It's always basically turning itself over. So what happens is osteoclasts, another type of bone cell, comes into this area in the osteon canal and begins resorbing the bone until it chews out an entire cavity. Then this cavity will have osteoclasts come in. Those are the bone-forming cells. Those bone-forming cells will line this cavity once the osteoclasts have finished resorbing bone, and they will begin laying down new matrix from the outside, and then they'll become encased in their own matrix, and more osteoclasts will come in and lay down another layer and another and another until you get back to having a brand new osteon and remodeled bone. So let's look at one more example of compact bone. This is the digit and nail slide, the developing finger and nail slide. Right over here in this area, you have some compact bone, but this is cut in the longitudinal orientation. Therefore, the osteons are not very apparent in this longitudinal organization. So let's take a quicker look at it. Here you can see that you have this solid bone, and this is compact bone, but the osteons are not very apparent because they're being cut in a different orientation. So as you look at this, you can find all of the osteocytes living within the bone. You may or may not find areas like this, which might have some osteoblasts lining the cavity, and there may in fact sometimes be osteoclasts in some of these areas, resorbing bone. These are fat cells here, and fat cells over here in the marrow cavity, inside the bone. Now let's take a look at spongy bone. When you look at these tibia slides, you should identify several different regions. In other words, the shaft of the long bone, what there is of it, is called the diaphysis, and the end portion here is called the epiphysis. You can find spongy bone both in the diaphysis, in the marrow cavity, and in the epiphysis, which has the spicules or trabecula of bone and marrow between them. So let's take a look at this, a little higher magnification. We're going to zoom in on the epiphysis. And here at medium magnification, you can see the trabecula of bone and all the marrow between them. Lining these bone trabecula are cells sitting on the surface of the bone. Those are the osteoblasts, bone-forming cells, and then the osteocytes inside the bone, which are trapped in there. Here at higher mag, you can see an osteoclast. This is a little hard to distinguish in this collagen-stained tibia, but there are several nuclei. This is a multinucleated cell. It's many times larger than the osteoblasts. Here they've kind of fallen off the bone surface. They would have been here. But you can see that they're much smaller cells than the osteoclast. Let's examine some spongy bone in this H&E tibia slide. I'm going to zero in the diaphysis region here, where you've got trabecula and lots of marrow around those trabecula. And here you can see a really nice example of a trabecula with an osteocyte inside of it, here, 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 and it's completely lined on this side and around on this side as well with osteoblasts. This area of the bone is undergoing a lot of remodeling, so you're going to see a lot of osteoclasts in this area. Even at this medium to low magnification, here's the trabecula we were just looking at, you can see an osteoclast here and another one here. Let's zoom in on those a bit and take a closer look. This one seems to have fallen off the bone a little bit, but that's actually good because here you can see what we call the Hauschip's lacuna. That's the trench, essentially, that this thing dug as it dissolved the bone. This is a bone remodeling cell, so it actually uses enzymes to dissolve the bone, both organic and inorganic matrix, and wear it down, break it down. Here's another one. This Hauschip's lacuna is a bit smaller, but that's a really nice example of an osteoclast sitting on a bone trabecula. Now let's discuss how bone is formed. There's two processes by which bone is formed. Using a cartilage template, that's called endochondral ossification, and de novo in a connective tissue space, which happens embryologically, that's called intramembranous ossification. Both spongy and compact bone can be formed by either of these processes. So the first example we have of endochondral ossification is using this developing finger and nail slide. Here is one of the finger bones. This starts out as all hyaline cartilage, this kind of dog bone looking shape here. And the first thing that happens is vasculature comes in and brings in bone-forming cells. They form this bony collar around the diaphysis region of this small bone. And as soon as that happens, the cartilage cells that used to live here die. This happens because cartilage has no vasculature inside of it. It's an avascular tissue, and it needs diffusion from the outside to the inside to live. So as soon as you put a bony collar around here, the chondrocytes die. But the vasculature also brings in bone-forming cells, and on the remnants of the cartilage matrix, where the cartilage cells used to be, then bone can use that as a template to form. So first it forms a template on the outside here, and then it starts using the cartilage matrix leftovers to lay down new bone on top of it. So let's zoom in on that and look at that process a little bit. So this is bone, and this is bone, and you can see this is cartilage. Looks just like you would expect hyaline cartilage to look like. But here, due to the ingrowth of bone, these cartilage cells are becoming hypertrophic, swelling up and dying. You can see their lacuna are pretty much got shriveled little cells inside of them, whereas these look more or less normal with normal matrix around them. But what's happening out here in this area that's completely cut off from diffusion? The bone cells come in and line the cartilage matrix, the pink stuff here, sit on top of it and lay down bone using the cartilage as a template. So that's endochondral ossification. This will proceed all the way down as this thing grows. The cartilage here is still growing because the fingers are still growing. This is not a particularly long bone, it's just a finger bone, but it does get a little bit bigger over time. So here we are back at the tibia slide again. This is a long bone, and when long bones get longer, they use this epiphyseal plate to do it. There's one of these at either end separating the epiphysis from the diaphysis of the bone. And this is a very specific area that has zones in it where the cartilage is doing different things. So you start out with this zone of resting cartilage or reserve cartilage. This is your stem cell population for the cartilage. These things begin to proliferate. So this is the zone of proliferation, and they do this in these stack-like arrangements. This is how the bone gets longer, by this proliferation of the cartilage. Then, the next stage is the zone of hypertrophy, where these cells swell up and begin to die. You can see these cells swelling up and then dying in this area, and you're beginning to see empty lacuna. In this area here, the cells have died, and you have what we call vascular invasion from the marrow cavity. This is the place at which bone cells are going to come in and sit down on this cartilage template. So where they're using this template to lay down bone, right here you can see bone being put down on top of this. This is the zone of calcified cartilage, because the bone cells come in and they calcify this cartilage matrix as they lay down bone on top of it. Even further up, you begin to see remodeling. So here you've got the purplish matrix that is from the leftover cartilage matrix, and then you've got bone matrix on top of it, the pink stuff. And then you've got remodeling going on. So the further you go up, away from the epiphyseal plate, the more remodeling you're going to see. And the pinker it's going to become, the less of this purple will be there. So eventually, all of this calcified cartilage matrix will be remodeled into bone matrix. So that's the zone of remodeling. So you have 5 zones in the epiphyseal plate, which is the resting cartilage, the zone of proliferation, zone of hypertrophy / cell death, zone of calcified cartilage, and the zone of remodeling. Now let's take a look at some examples of intramembranous ossification. And in order to do that, you have to have embryonic tissue. So here we are back at the pig embryo slide to do that. At low magnification, you can see these bone trabeculae here and all in this area here. All of this is formed by intramembranous ossification. And really what's happening here is you have a undifferentiated connective tissue space, which is mesenchymal cells. And you can see those back here in the background. And in that space, cells receive signals to differentiate into bone-forming cells. And they begin to differentiate into bone-forming cells and to gather into little clusters like this. And then, as they further their differentiation, they begin to make matrix. And eventually, that matrix gets calcified and becomes bone. At this point, you could call these cells on the outside osteoblasts. They are differentiated. And here is a little bit further along. So here you have osteoblasts that were differentiated from this mesenchyme out here. This pink stuff, which is the uncalcified bone matrix, osteoid, if that's called. And then you have a bone trabecula in the middle, which is calcified, with bone cells trapped inside, which are osteocytes. This whole area up here that I pointed out at low magnification is just further along in the process than this little area down here. More trabecula are forming down here by intramembranous ossification. These are already formed, and in some cases are remodeling. So here's a really nice example of an osteoclast remodeling this bone trabecula right here. Here's the Hauschip's lacuna where it's chewing away at this bone, and you can see the multinucleated large osteoclast cell here sitting on the bone trabecula. And then the cells, of course, that are covering the bone surface are osteoblasts, these guys. The slide of the spinal column is a really nice slide for doing a little bit of a review of some of the tissues we've looked at. You can see examples here of spongy bone in many places here, all around here. That's all spongy bone. Then you can see examples of hyaline cartilage here, the dark areas here, and then transitioning here between this hyaline cartilage and this dense connective tissue here, there's going to be fibrocartilage. So let's zoom in on this area and look at all of those and get a kind of a review of this whole thing. So here's trabecula. These are all bone trabecula. You've got calcified cartilage in the center, bone laid down on that template. So this was endochondral ossification here. Then you've got the cartilage where this originally came from, still out here. This is hyaline cartilage. These are becoming hypertrophic as this grows. And then transitioning outward from this hyaline cartilage, you can see this area here with the lines of chondrocytes and the big ropey looking collagen type 1 fibrils. So this would be an area of fibrocartilage right here. Transitioning between this hyaline cartilage here and this dense regular connective tissue here, which is the covering on the outside. If I zoom back out, you see there's a lot of other tissue here. This stuff out here is all muscle, and we have yet to examine this skeletal muscle in great detail, but we'll do that in a couple of labs down the road. At lower magnification, you can kind of go around this whole thing and see other examples. Here's more cartilage down here with bone forming in the middle. Let's zoom up, and you can see there's marrow and bone and hyaline cartilage on the outside there, because bone is going to take over this whole compartment. Finally, the last slide we're going to look at in this lab is the parathyroid gland. This isn't bone or cartilage. However, it's a very important tissue for the endocrine control of calcium, which comes from the bone. So the parathyroid glands, this is just a portion of a section of one of them, are considered vital organs because they're very important in maintaining blood calcium levels. There are two main cell types here in the gland, in the parenchyma of the gland, I should say, because there's connective tissue that keeps it all together and vasculature, which forms the stroma of the gland. But if you zoom in just a bit here, you're going to see these small dark cells, which are called chief cells. These are the cells that produce parathyroid hormone, which increases blood calcium. And it does that by stimulating the release of calcium from bone via increased activity of the osteoclasts. The other cell type, if I can get this to zoom properly, is called an oxyphil. So those chief cells are the dark ones. You have these areas where they're kind of more eosinophilic and light, these areas here. So here's chief cells all around, and these areas where you have these lighter cells, eosinophilic looking, more separated. These are the oxyphils. The function of oxyphils is not really well understood. They're thought to be maybe a regressive form of the chief cells, or maybe a quiescent form that's just resting, but it's not really known. So they don't have any functional significance for you, just so that you know there's two different cell types here. But really the chief cells are the ones, the heavy lifters. They're doing the work of this organ. So that's all we've got for today, and I hope this was a useful medium for you to go through your lab.